fairly new, and that it might be more useful to talk in a couple of years. But actually, the organizers were fairly welcoming. I'm going to talk about emerging nanosensor technologies for rapid label-free glycoprofiling. And I lead a research group of about 26 postdocs and graduate students at MIT. I'm a chemical engineer by training, and we focus on nanotechnology. And part of my group, we make new kinds of nanosensors that are very, very sensitive, and we work on molecular recognition. And I became interested in the problem of glycan detection and glycoprofiling and carbohydrates because of its longstanding difficulty. So I consider it a very challenging analytical problem, and that's really why I'm motivated to come to it. So I don't have completed stories to tell you, like the previous talks were very elegant and have gotten to a location. I want to just talk about concepts that'll be very interesting. So one is I'm very interested in new ideas that we can use for multiplexed, label-free glycoprofiling arrays for nanosensors, using nanosensors, and I'll talk to you about what ideas we have there. And then I would like to talk to you also about a technology we developed at MIT that makes essentially synthetic molecular recognition sites, kind of like synthetic antibodies, or in this case, maybe we can make synthetic lectins, but they're nanoparticle templated, but they're made of essentially synthetic polymers, and it's my hope that they could help significantly with this problem. And then I have one slide where we're making nanosensor platforms that operate quite well in vivo, particularly in a murine model. And I would like to introduce the idea that we could monitor glycoprotein biomarkers in vivo in real time. Okay, so just moving to this first topic. So one way to think about it, in fact, the previous talk from Children's really set this up nicely. So the extensive analytical suite of techniques that you need now to actually glycoprofile a particular specimen is fairly extensive. There's a huge technological need to make a rapid platform, not just in the research space or for clinical. You can also think about the pharmaceutical industry. So it's very, very clear that from a manufacturing standpoint, the extent and type of glycosylation that you observe during manufacture is a control variable. It's something that can vary, and it's been clearly linked to potency here. If you want to take a look at it, and but we can also think about new rapid diagnostics that can very quickly and at very low cost profile aberrant glycosylation. And so where I work is in this space. So if we rank all types of nanosensor technologies, I think the battery on this is done. It's no problem. If we take a look at all emerging sensor technologies, from traditional microarrays, cantilevers, different biophysical techniques, we can rank them in terms of how many different multiplex sites, if you want to think about that, how many different aspects of the analyte mixture can be assayed at once, and then sensitivity. And nanosensors have this ability, because they're very small in profile, they benefit from the convergence of two different aspects. They can get sensitivity better than femtogram per mil. In fact, down to single molecules. I won't show that in this talk. And we can multiplex them rather significantly. So they can carve out this space of where we can get very sensitive detection. And then also massively multiplex that, and also at very, very, very fast assay time. And we've been thinking about the traditional lectin microarray and the information that's lost when essentially you're just looking at the strongest of what are already very promiscuous binding and also weak. So this is our first new idea, and we welcome feedback from the community. So I'm an engineer. If you start to think about, we started to formulate this glycoprofiling problem as a very simple matrix algebra formulation, and I will not go through the mathematics for this community here, but it's very simple here. We can think about a glycan sample using this matrix P, right? Which essentially the rows here are each of the different, say, protein isoforms that would be in our sample. And the columns here would represent the different glycans that are displayed. Okay, so I just have an example here, five different glycans. So then finding this matrix P, which essentially identifies the types of carbohydrate chains that are displayed on the protein isoform. Essentially, mathematically, if we find P, then essentially we know everything about the glycosylation of our sample. And then we can couple this to, we have a concentration for each of these isoforms, so then we can actually form a vector G. This is the concentration of our different glycans in our mixture. And actually, for someone like myself, it's very useful to think about the glycosylation, the glycoprofiling problem in this way, because then I can write simple equations on how a label-free lectin microarray should behave. This is like very simple, just reversible binding to an array element theta. Say our sensor is able to detect some glycan J bound to lectin L. I can just write a very simple site balance for my sensor, and then I can use not just whether something has bound after I've washed, but I can use the dynamics to actually extract a lot of information that can give us better orthogonality. So the rate at which something binds and also the dissociation constant are in addition to whether the extent to which it's bound. We have a very detailed theory. This is just for an example, assuming monovalent interaction. We saw from a previous talk, polyvalent interactions are very important for these systems, and it's just it just means that there's a system of equations with a sum. And so this is the approach that we're taking. And I'll tell you a little bit about the nanosensor technology before I show you some of the data that we have here. So we're using, for this particular problem, we're using single-walled carbon nanotubes. They fluoresce in the near infrared. They're semiconductor nanoparticles. They have near infrared fluorescence, and this is an excitation profile here. So I'm exciting them with different wavelengths of light, and they're shining near infrared light at different wavelengths, and there are different flavors here. We can talk about what causes these different flavors, but they're non-photobleaching, so we can make an array that essentially can last. It's a fluorophore, but it can last for a very long time. There's no irreversible photobleaching. This is indocyanine green. It's a near-infrared a dye that's used for tissue spectroscopy. You can see that it photobleaches, but the carbon nanotube does not photobleach. And this is a technology that we developed at MIT. It solves a very important problem in nanosensor space. It allows us to detect the binding of macromolecules very selectively, but avoid things like non-specific absorption. And what we do is we take the nanosensor and we put it into a hydrogel that's very resistant to protein absorption, and then we use this. We use a nickel chelator chemistry to bind a divalent ion like a cupric ion, which acts as a proximity quencher to the tube. Then we can dock, say, a histag lectin, and anything that interacts with that histag lectin, we can then transduce to the nanotube down to the single molecule level. So it's a very sensitive assay. And we have used this recently. This just demonstrates the analytical capabilities of this approach. This is not for glycoprofiling, but we looked at a famous example in the analytical literature, IgG recognition by protein A. Protein A is used ubiquitously, to bind and detect this FC region of the IgG. And it's confounded other biosensor platforms because because of this multi-polyvalency interaction. So if you try to measure an effective dissociation constant, as some have here, these are from the literature, from inframetric sensors, acoustic wave. So they find confusing ranges of dissociation constant, and it's because it's not a monovalent interaction. And so here we show the black is our curve. We are able to get data accurate with enough precision to actually deduce the precise bivalent binding kinetics and equilibria of IgG protein A. So this just came out in analytical chemistry. So, but it highlights that our platform is able to nuance out this kind of binding interaction. And as a control, if you take a look, if you just look at human growth hormone binding to its receptor, that's exactly monovalent. So it's a monovalent. So you're supposed to see that we can distinguish those two. Okay, so this is the technology that we're working on here. This is generation 1 is a single measurement at a time. Generation 2 is 4 times a 96 well plate. It's completely automated. And generation 3 we're developing in parallel is 4,000 nanosensor spots. And we eventually want to go to the 4,000 spots in the 96 well plate. So each well plate will have 4,000 spots. So that gives you a sense of the technology. These are our results so far. So this is just looking at human, mouse, and chicken IgG, and just with six commercially available his-tag lectins, and we have many more on the way. But some of the things that I want to point out is that we can get a lot more information from this heat map. So so this is a traditional heat map of what has bound and what has not bound, right? So you see, for example, the chicken has high mannose binding lectin affinity, but then you can also map from each sensor spot. You can get the KD, a lot of other information as well. And you're supposed to see here that whereas just looking at what the extent to something has bound, you see a lot of blue here, but we can actually nuance and get a lot of orthogonality if we look at KD, and potentially also binding kinetics and so forth. Okay, and you're supposed to see here that what we're detecting so far is fairly reasonable. We see interactions here. They're largely reflected in the KD, but what I wanted to highlight is that we're able to get positive information on even weaker binding interactions. So you're supposed to see in here that it's the start of profiling weaker binding interactions. And I hope I'm not grinding gears, but I wanted to stop there, and I wanted to introduce this idea of Kaufmore, because I wanted to stimulate some discussion here. So the idea here is to switch gears. Do we have to use lectins, either recombinant or natural lectins? Are there new ideas for binding glycoproteins in here? And so here I just highlight that lectins essentially are nature gives us these motifs for molecular recognition, normally a biological polymer that is constrained in some three-dimensional space, and we can get recognition if we have amino acids. We can get the antigen binding domain of say an antibody. We can use DNA or RNA. We can use nucleic acids for aptamers, and then that's essentially it as far as what nature gives us for generic molecular recognition platforms. There is a synthetic platform called molecular imprinting. It makes essentially an adsorbent for a molecule. It has an advantage that it can be used with synthetic polymers, but it normally targets low molecular weight species and doesn't have the broad applicability of say the use of antibodies or aptamers. And so we developed a platform for synthetic molecular recognition at MIT. It's called Kaufmore. We published this at the end of 2013 in Nature Nanotechnology. The idea is that you take a nanoparticle, and the nanoparticle can help shape and template a synthetic polymer, which you pick out from a library of such polymers. So you use forces like the following. If I have a carbon nanotube, it's hydrophobic. It doesn't want to be in water. But if I have a polymer that's an amphiphile, the polymer will absorb onto the surface. It'll put the hydrophilic portion into the water. The hydrophobic part will stick to the surface. This makes what's called a corona. So almost all nanoparticles in solution have some kind of polymer or surfactant capping or corona. And the misconception has been that these corona phases are essentially disordered. But if you screen a library of polymers, you'll find really exquisite molecular recognition with just certain compositions of polymers adsorbed to the surface. And so this is kind of like finding an amino acid sequence that happens to fold into a structure that later becomes your enzyme or your antigen binding domain. And so there are a lot of examples of Coffmore in the literature now. So if you take a look, so they're really surprising, in fact. So this is just a Ritzy-Peg-Ritzy polymer. When it maps to the cylindrical nanoparticle, it makes these loop structures. So this type of loop structure will recognize estradiol, both alpha and beta, really exquisitely. But if you mutate the feet to some other to this FITC, your selectivity goes away and it looks a lot like a lipid peg. Right? So this structure, we call this a Coffmore structure, a corona phase molecular recognition. It can be very, very selective. And this just shows you an example applied to neurotransmitters. We published this in JACS last year. This was just a postdoc doc of mine did a screen for some common neurotransmitters, and this gives you a sense of what a Coffmore screen looks like. These are different polymer corona phases, and you can see here that we see this turn on. Red is when the emission of the fluorescent sensor is getting bright. We see a turn on sensor for dopamine, and it has some interference with epinephrine, but you can see here that there are some corona phases where there's no response, but you're supposed to be seeing here that the corona phase plays a very big role in what is selective here. I want to point out that this automatically the process makes an exquisitely sensitive nanosensor, a single nanoparticle that can recognize the molecule here. You can use it right away. So you can see here, this is on a microscope slide. This is an array of nanosensors, and Sebastian has put some dopamine fluid, and you see this bright response here. It's an exquisite. It's actually the first fluorescent probe for dopamine. Nobody has ever made a fluorescent a fluorophore that actually acts as a dopamine indicator. I was very surprised about that. You can see down here that it's reversible. And what does this have to do with carbohydrate rate recognition? Well, I have another student, Ji-Young An, and she is tackling this. She's applying Coffmore to carbohydrate recognition. And what we found so far is very surprising. This is a very powerful technology. She's been making these polyacrylic acid-based Coffmore backbones. She uses a diborotic acid. She uses a single borotic acid on this aromatic ring. The aromatic ring should stack onto the hydrophobic nanotube, and she's able to distinguish. I don't know if this is impressive to this community or not. She's able to actually distinguish these sugar alcohols. If you take a look at this, these are all 6-carbon sugar alcohols. They differ only by just the cis-trans display of these OH groups. This is a very, very difficult problem. I don't think there is a synthetic human-made system that can do this distinguishing. But this Coffmore, she found a Coffmore phase where if she puts the borotic acid in the meta position, she can actually distinguish these quite well. And you see one thing here is that between para, meta, and ortho, the composition of the polymer really plays an enormous role into the structure of that corona phase. And so she also showed that there's a polymer length dependence here, and we haven't published this yet, but we're working on it. So the polymer above a certain length can do this recognition. So I hold out a lot of hope that we'll be able to make new structures that can do this, that can be applied to this challenging problem. I don't know how much time I have. Maybe I have a few minutes or. Okay, so then the question is, can you detect glycoproteins, whole proteins, not just small molecules? And I have a postdoctoral student, Gilly Basker, and she started to apply Cough-More to. She wanted to look at fibrinogen. So fibrinogen is a glycoprotein. It's found in whole blood. What she did was she took the most abundant proteins of whole blood and made a library of analytes, and we're using that to screen against for our molecular recognition. And this is one of her Cough-More screens. These are different corona phases, and she found this really nice phase. It's exquisitely sensitive to fibrinogen. So actually, this kind of shows you it's really kind of a synthetic antibody, if you will. It's able to pick out fibrinogen from these other what are otherwise very, very, very sticky molecules, including albumin and so forth. And this was our first example of protein Cough-More ever. So actually, we decided to study it just to try to understand this phase. It's pretty neat. Fibrinogen has these two D domains flanking this middle E domain. And actually what we did in a series of controls, we looked at cleaved portions versus intact. We found that the recognition side, it's not recognizing carbohydrates, but it's recognizing this D domain. And it recognizes both of them. And there's a lot of agreement here. If you take a look, you can make a kinetic model where it binds one nodule, then the second one binds, and you get really nice kinetics. These are all different kinds of nanosensors, all different flavors of carbon nanotube nanosensors here. But you see here, you get nice equilibrium binding here with the surface. And this, I think, I will skip. I'll skip over. Well, this just shows that again, you can make nanosensors on a surface. You can actually these are immobilized nanosensors on a microscope slide just at room temperature. And you can see here, if you flow fibrinogen over, you get a very nice response. They're able to kind of pick out the fibrinogen and you get and recognize it. I only have one slide left on the last topic. I don't have enough time in a 20-minute talk to go through all of it, but I wanted to advertise that we're working on a really interesting nanosensor platform. It's kind of a hydrogel tattoo. And I'm interested in being able to measure macromolecules in real time in vivo. I think that this will be useful for animal research, and we hope to apply it to humans at some point. But it's so this is the MIT Smart Tattoo technology. We've made a lot of progress on it. The idea is that you can take a passively excited hydrogel fluorescent beads that contain nanosensors. We can use Cough-More for molecular recognition, and we can implant these subcutaneously, and then they'll fluoresce in the near infrared. Your body and thick tissue tend to be transparent to the near infrared. You can get about 4 to 5 centimeters of near infrared light. I have a lot of data on this, but the idea is that we could implant this platform. Here, this is in a hairless mouse model. That this is just these beads injected with the syringe. They're very, very stable. This is after 26 days from injection. In our paper in Nature Nanotechnology, we show that sensor platforms like this can be viable more than 400 days in a murine model. We did an A129 mouse. It's the longest I think any sensor platform has ever operated in vivo. And if you know of a system that has been demonstrated more than a year, then I would like to see that. What single sensor, no recalibration, but just an implant. So right now we're applying this to glucose, to insulin, and I've just started a project. Gilly is going to start to apply these to things like protein biomarkers, where we'll be able to detect them in vivo. And with that, I went over my time a little bit. With that, I'd like to thank my group and like to thank these funding sources. I'm happy to answer any questions. Thanks.
Really interesting. Yeah, sure. Some questions maybe over there first.
Is it? Oh, yes, super. You say that these sensors are highly sensitive, but how well do they react to noise? So if the samples are more complex, is that a problem for them? Or
So the sample complexity is the molecular recognition problem, and that's why I've turned my attention in the past 2 years to making molecular recognition platforms. So as of maybe 4 to 5 years ago, you started to see in nanotechnology platforms that essentially can go down to single molecules. So they can take a bulk mixture, and if there's a molecule that you want, to detect inside the mixture, as long as it can make it to the sensor, it doesn't say how long it takes, right? It could take a million years, right? Or if the sample's adsorbed. But we've essentially broken the sensitivity barrier. Okay, so sensitive. So your question about complex mixture is one of molecular recognition, and so the question is, what are you trying to detect, and can you detect molecule A versus background? So I think what I showed you were some new molecular recognition platforms. We're detecting fibrinogen out of whole blood, so that's about as complex. And the in vivo work also has an environment that's complex. But I think I'm answering your question, right? That it's a selectivity and sensitivity problem separately, if that makes sense. Did you have a question?
Yeah, Neil Price of the USDA. I think. So smart tattoos, is that a contradiction in terms, perhaps? I was wondering about the availability of these nanotubes. Where do you actually get them from?
So we grow them in our own laboratory, but there are suppliers that mass produce them now, and the field has gotten very good now at purifying and getting them to the level of a single chemical reagent. So there's a company in Westwood now that produces them in bulk by flame synthesis. They've actually licensed a lot of our patents. Sigma Aldrich sells them too. So if you wanted to do experiments, most of the time a supplier will send you raw material. You can think of it as like cell lysate if you want, and you have to do your own protocol to separate them out to get pure species. That's changing now. Some companies now will sell you very highly pure material. So if that makes sense. Does that answer your question?
Can I ask another?
Yeah.
So I was very interested about the work you were doing with the polyols there.
Yeah, so was I.
Yeah, extremely interesting.
Yeah.
So can you distinguish, do you think you can distinguish D and L polyols? Or have you looked at a rabinitol and mannitol, for example?
Yeah, so we are seeing, we are discovering, yeah, so I have a lot more about that. We can talk about it at the break. We're seeing Kaufman phases that are pretty surprising. So we can see, in addition to those, we found a Kaufman phase. I have some extra slides on it. We can see, it's a Kaufman phase that it can detect sucrose, but it doesn't respond to glucose or fructose. So actually, we think we can see that glycosidic bond actually, and we can look at different structures. We found a lot of cases like that where we can see very particular structures displayed from the carbohydrate chain, and that's getting at what you're talking about, right? And so we're very interested in scaling up and really. Producing a suite of Kaufman phases that can recognize very particular chemical motifs. But we're looking to partner with people who have expertise to try to understand what's important for this community, to detect which parts. But so thanks for your encouragement. Yeah,
Fine, we're good.
Yeah, is your question?
Very nice. My research is also involved in chemical engineering of delivery systems. In particular, looking at hydrophobic chemotherapeutic drugs. Have you looked at some of these nanotubes or carbon tubes in terms of encapsulating the chemotherapeutics in the release profiles?
Yeah, so that's a good question. I haven't, but there are colleagues of mine who are. So there are large diameter carbon nanotubes. The ones that I work with are quite small. So normally, yeah. So the ones that are involved in this research are between say 1 to 3 nanometers. It's possible that you could. It's possible to encapsulate materials inside of them. Depending upon how long they are, it may take a while for the molecules to diffuse outward. There's some really nice work from Lawrence Livermore National Labs that have actually used really short carbon nanotubes as prosthetic ion channels. So believe it or not, in fact, we debated this in the field for quite a while. A very small nanotube will actually dock into a lipid bilayer membrane and will actually allow ions to travel, outside of a cell. Anyway, go ahead.
I guess the issue here in terms of the literature is these carbon nanotubes do develop toxicity in the in vivo animal. And also the other efficacy of these delivery systems is that does it hit the target?
Yeah. So actually, I'm glad you brought that up. Actually, since we started our animal work after 2 or 3 years, fewer people ask that question now because I normally in a longer talk I show lots and lots of animal data. Yeah. So nanotoxicity is something I actually teach a module at MIT on it. The long and short of it is that in the early days of nanotoxicity, when traditional toxicologists were kind of applying their tools to the nano realm, there was a lot of confusion, a lot of lumping material together. So if you look in the literature, you'll find in vitro toxicity studies of gold nanoparticles of the same size and shape that are actually were FDA approved for as use of a therapeutic. Right. Long and short of it is the toxicity of a nanoparticle is dependent upon size, shape, and the chemistry that it displays. Yeah. So there are kind of flavors of nanoparticles that are quite toxic, and then it's an engineering problem to make a particle non-toxic. Interestingly enough, in our Nature Nanotechnology paper, we did several studies. I mean, one we did these hydrogel implants where the nanoparticles were safely encapsulated in the hydrogel. But actually, we also looked at formulations that we injected into the mouse via tail vein, and they accumulated into the liver. We actually used them as sensors in the liver, but they cleared the lungs and so forth. So we looked at biodistribution, and you're seeing that in the literature. So you'll see that there are carbon systems that people are interested in for delivery purposes, and those appear, at least the acute toxicity seems to be minimal. I think long term we have to figure that out. But in general, across the nanotechnology space, it's the chemistry displayed, or whether the molecule the particle can leach toxic molecules. Or does this make sense?
Do you think the glycosylation that you're putting on the nanoparticles alleviates the toxicity?
Well, that's a good question. So in our case. So, in our case, we're making something like an implant and we've done a lot of measurements to show that the nanoparticles are not leaching from the area of the implant. I think it's possible to make a hydrogel that will contain the nanoparticles. So we haven't done a lot of work on that particular question. We're not essentially focused on delivery, but there is a part of the community that is. And I can follow up and tell you what I know and so forth, and we can talk more about that. Okay, excellent, good. Yeah, thank you very much.
I have about 100 questions, but I think we better move on. Let's thank Michael again for a really, really interesting talk.
Thank you for your interest. Thank you.